In the previous lessons, we have learned the difference between descriptive, correlational, and experimental research methods. We also discussed the three major types of research, namely uh, laboratory, field, and lastly, the natural experiment. In this lesson, we will learn some of the terminologies we use in experimental research. But before that, this presentation is ready to download, so make sure to share it with your friends and classmates if you find this material helpful. Let's start with the discussion of different variables. Okay, now we have dependent, independent, and extraneous variables. Let's define each one. Dependent variable is the one that is influenced by the independent variable or the intervention. So basically, the criterion by which the results of the experiment are judged. Okay. So in contrast, the independent variable, it is the one that is being manipulated by the researcher. So independently, this variable is trying to influence the dependent variable. So it is hypothesized to be the cause or the causal influence of the effect. Okay. On the other hand, we have the extraneous variables. Extraneous variables are different with independent variable. So basically, extraneous variables are referring to those variables that really affect the results of the dependent variable. For example, researchers usually control all or almost all of the factors or extraneous variables whenever possible. One form of these extraneous variables is confounding variable. Okay, so confounding variable could be a uncontrolled extraneous variable that likely affect again the results of the dependent variable. Okay, so let's discuss extraneous variable more deeply. Okay, so basically extraneous variables has different um, factors or influence. So for example, we have the history effect, the cohort or the group effect. We also have the maturation effect, the testing effect, as well as the instrumentation, how we use the instrument, and of course the selection effect or the sampling bias that we have, and of course the mortality or the sample attrition. So to control these extraneous variables, we're considering blinding. So blinding is a technique we use or the researcher use to control subjects' knowledge or whether or not they are have been given the experimental treatment. Okay, so we have different forms of blinding. We have the single blind study and the double blind study. The single blind refers to the subject as being unaware as to which group they are in. So while the researcher knows which subjects are in each group. So for example, we have these particular subjects and they don't know if whether they are in a experiment or a control group. Okay, so in a double blind study, both researchers and the subjects are blind to group assignment. So by doing so, we can control or lessen the impact of this effect to the experiment. Okay, so we have the placebo effect. Placebo effect occurs when uh, subjects expectations or beliefs influence or being determined by the experience in a given situation. So in other words, simply expecting something to happen even there is really nothing about that treatment. So for example, giving a pill which really do not have something in it, so it is a placebo. So it might affect the way the participant may behave just just because to know uh, the real effect of this experiment. Okay, now let's go with the experimental hypothesis. When we say experimental hypothesis, we are we want to know whether there is a significant difference between the treatment versus a non-treatment, or both treatment, or both groups, the experimental and the control groups. Okay, so in order to conduct an experiment, a researcher must have a specific hypothesis to be tested. Okay, hypothesis can be formulated either through direct observation of the real world or after careful review of previous researches. For example, a researcher might want to think that aspiring psychometricians or professionals are more effective in terms of self-study versus attending group study. So if the researcher thinks in such a way, then basically that researcher might formulate or create a hypothesis. So basically it might be self-review, increases one's efficacy to answering mock tests. So to determine this hypothesis or to test this hypothesis, the researcher may conduct an experiment. Okay, so we also have the operational definition. When we say that operational definition, it is a description of how we measure various variables. Okay, and it is important in allowing others to understand exactly how and what researcher measures in a particular experiment. So the researcher will define specific variables in that study, basically on how he or she define those variables. We also have the ecological validity. In the previous lesson, we have learned about ecological validity. Particularly, it's used to the real world. So basically, ecological validity refers to the level that an experiment can portray real life events. Okay, so on the other hand, we have the manipulation. When we say manipulation, okay, it is the experimental treatment. So basically it is direct or indirect. So manipulation refers to independent variable being studied. When we say direct, it is referring to modifying the physical setting. On the other hand, indirect refers to changing the verbal instructions to indirectly manipulate the independent variable. Let me explain this. For example, a researcher may want to know the stress level of subjects, which is impossible to manipulate the stress or measure it directly. So the researcher can indirectly stimulate this stress tendency by means of verbal instructions, or telling them you are given only 3 minutes to prepare for a short presentation in front of the class. In that case, a researcher is trying to induce the stress among subjects. Okay, so manipulation check to know if this has effect to the dependent variable. It could be measured through a survey or questionnaire. Okay, when we say conditions, okay, it is either experimental or control groups. Okay, this is different levels of independent variables. For an instance, we are the most basic experimental design involves two groups, or the experimental group and the control group, whereas the experimental group will have this experience of the treatment. Since experimental administration is the only difference between the control group and the experimental groups, a researcher can secure that any differences between such conditions are due to experimental manipulation or the intervention itself rather than chance. Okay, so let's discuss the experimental group and the control group. Again, the experimental group is exposed to intervention, such as set of subjects will undergo uh, training, for example. The control group, on the other hand, will not receive or will not be exposed to experimental intervention. For example, set of subjects who will not receive training. Then later on, the control group's performance as well as the control group's performance, their post-test could probably be measured, or their pre-test will be measured as well. Okay, so when we say measurement, it could be in a form of t-test or analysis of variance. Okay, so it will be compared to the experimental group. Thus, to know if the training is likely increase the work performance, for example, of selected subjects. So the null hypothesis for this can be training does not change the work performance of the employees or the subjects, or the alternative hypothesis can be that training can really increase the work performance of these subjects. Okay, we also have different experimental distribution. Okay, when we say experimental distribution, we have within subject and between subject. When we say within subject, it refers to repeated measures. Okay, again, it is basically repeated measures for related samples. When we say related samples, subjects are tested under all treatment or conditions and explore the within subject designs as the choice of distribution in order to one, we use within subject for us to minimize the impact of individual differences. Individual differences is very important because the researcher will mostly observe the same set of people under two conditions. If on the other hand, between subject is more prone to these individual differences, which likely influence the result or the dependent variable later on. So another reason why we use within subject is for us to save cost, as between subject design is almost always require a larger sample compared to within subject design. Okay, also within subject design is likely prone to order effect. When we say order effect, this occurs if the experimental method requires that the same subjects will be exposed to two or more experimental treatments. So this error may occur due to the order in which the treatments are presented. To avoid this one, we use counterbalancing. When we say counterbalancing, it is referring to the half of the subjects exposed to treatment A and the half of the subject treatment B. Okay, another half will expose to treatment B first, then treatment A. So this will likely to eliminate the effects of the order presentation. Let's discuss the between subject design. When we say between subject design or distribution, it refers to the independent measures or the unrelated subjects. Basically, there are two different set of individuals or subjects. So subjects are tested with only one treatment or condition. So set of individual A can be for the experimental condition, and the other one is for the treatment or the unexposed groups. So this distribution is prone again to individual differences. Okay, it is also prone to systematic error. When we say systematic error, it refers to an error that occurs in the same experimental condition. Every time that the basic experiment is repeated, a systematic bias may occur. For example, the experimental groups always receive the treatment in the morning, or control groups always receive the treatment, or control groups are always in the afternoon. So this introduces an uncontrolled extraneous variable, so a time of a day. The following are the sources of this error. We have the demand characteristics, the experimental bias effect, and the Gini effect. Let's discuss this one by one. When we say when we say the demand effect, so experimental design, procedures or situational aspects of the experiment that provide unintentional hints to subjects about the experimenter's hypothesis. If this occurs, subject likely to act in a manner consistent with the experimental treatment. Okay, so the most prominent demand characteristic in the person actually administering the experimental treatment. And experimenter bias effect, it refers to the subject's behavior caused by experimenter's presence, actions, or comments. So it can be accidental. Okay, so the Gini effect, on the other hand, it is referring to the experimental results caused by the subject's changing normal behavior or attitudes to cooperate with the experimenter. So in order to avoid systematic errors, we try to use the randomization. Okay, so this one. Randomization is very important with experimental study. Why is that? So basically, it is the assignment of subjects and treatments to groups and is based on chance. Okay, so this method lessens the impact of individual differences which will be assigned to experimental or controlled conditions. Now, a researcher may use random number generator via Microsoft Excel to use this randomization. So this is not a random sampling, okay? So randomization can still use convenience sampling, but randomly assign those individuals or subjects to different conditions. For example, we have for the experimental group, we have for the other one or the untreated groups, okay? So usually it can be found in true experimental designs, which will be discussed in the next video, okay? So random assignment again allows us to assume that the groups are identical with respect to all variables except the experimental treatment. Okay. Now we also have the survey research. When we say survey research, it is used to learn about the characteristics of the subjects. When we say survey research, we can consider longitudinal or the cross-sectional. Okay. Basically, survey research helps us to an experiment to describe the population. This can be explained by longitudinal again. The longitudinal is conducted several points in time. So this is mostly applicable for group studies or cohort studies to track the same group of people. So there are types of longitudinal studies. We have cohort studies and the panel studies. When we say cohort studies, it tracks the lives of groups of subjects who undergo the same life conditions within a given time period. So basically, cohort studies represent a particular group of people rather than population as a whole. Again, then we have the panel studies. When we say panel studies, it follow the same individuals over time. Okay, again, cohort, group, panel, individuals. Okay, so basically these studies aim to remain representative of the population of household as a whole. For example, so these surveys need to ensure that they can include new persons or families in from coming from the said population or sample. Okay, we also have the cross-sectional studies. When we say cross-sectional studies, it is conducted only one point in time. In contrast with longitudinal, it is multiple times. Okay, so cross-sectional is only one point in time. So this can only be conducted to different individuals with the same characteristics which are being compared. Okay, when we say sample size, it can be gathered through a rule of thumb that per condition, for example, we have the experimental and control group. For experimental group, we can assign 20 per that condition, and another for control group, which is another 20. Okay, so when we say sample size, we can use the G power as a tool to know whether it is a the right sample size. Okay, so G power is an open source statistical power analysis. So it is intended to examine or analyze various classes of power. Okay, when we say power, again, it is the capacity of an experiment to reject the null hypothesis. So it is basically from the chances of detecting the effect of the um, independent variable to the dependent variable. Basically, it ranges from 0 to 1. So the smaller the effect sizes can detect, the more it is powerful. So to increase the power, I think we can consider increasing the number of the subjects, or we use within subject design to avoid the impact of individual differences. We can also increase the level of significance at 0.1% instead of 0.05%. Okay. Now, we will discuss the experimental designs for the next video. I hope that you have learned a lot in this presentation. And again, this presentation is available in our description given in this video. Okay, thank you so much, guys, and we will see you in the next video.